Good morning, everyone. Welcome to Lawrence Livermore National Lab's Virtual Science on Saturday series. I'm Dr. Joanna Albala. I'm the manager of the Science Education Program at the lab, and this is our second lecture in this year's Science on Saturday series, themed Energy and the Environment. We're excited to share the many ways in which the lab is doing cutting-edge research in different areas relevant to our planet. Now, before we get started, I have some housekeeping instructions. Depending on your device, you might want to go to the top right-hand corner on the slides and you'll find a little screen icon. And if you click on the side-by-side view, this will maximize the slides and the speaker view to the right of the slides. You've entered the event muted with the cameras off and you'll not be able to cross-chat. But if you have questions, you can enter them in the question and answer box, and we'll try to get to as many of them as possible at the end of the presentation. Also, you may look like you're the only one here, but trust me, there are many, many others in the audience on the call. I also want to acknowledge that we will be recording the presentation today, so no outside recording, please, or screenshots are permitted. We'll also make the recordings available on the Science Education website at the end of the series this spring. Finally, before I introduce our speakers today, I'd like to thank you for taking the poll, and it looks like most of you are from Livermore today, but thank you all for joining us for today's presentation. And now I'd like you to meet our speakers. Xavier Malali received his high school diploma from Berkeley and then went on to get a BA in Environmental Studies from UC Berkeley and an MS in Marine Biology from the College of Charleston, and then his PhD in Oceanography from the Scripps Institution of Oceanography at San Diego. Lots of California schools in his background. He's currently a deputy group leader in the Environmental Isotope Systems Group in the Nuclear and Chemical Science Division at Lawrence Livermore Lab. Ty Samo started his education at Vacaville High School, and then he went on to get an associate's degree at Solano Community College, and then a BS in cell biology and biochemistry from UC San Diego, and an MS and PhD degree in marine biology, also from the Scripps Institution of Oceanography at UC San Diego, and he's a staff scientist in the Environmental Isotope Systems Group in the Nuclear and Chemical Science Division at the laboratory. And joining them today is Ms. Erin McKay, who's a biology teacher at Tracy High School. She received her BS in biology with an emphasis in plant biology and her science teaching credential from the University of California at Davis. And we have a whole group of presenters today with a strong education in our California public school system. She is a teacher at Tracy High for almost the past 20 years and also teaches in our biosciences program at the laboratory. And now, without further ado, let's get started.
Thank you, Joanna. Ty and I are really excited to talk with you today about algae. Hopefully, you will have just as much fun as we will this morning. So let's get on to it. We have three main goals for you to take away from today. The first is that algae are really amazing creatures that are incredibly important for the global climate and global carbon cycling. And so we'll tell you a little bit about that. The second thing we would like you to take away is that we can actually use algae to fix some of our major environmental problems, including climate change. So we'll tell you about that in the middle of our presentation. And finally, we want to talk about the algal microbiome. We have realized that microbiomes are important for the health of all organisms, not just humans. You may have heard of the human microbiome. Algae have their own microbiomes as well. And the microbiomes are the microbes that are associated with the particular host and that keep them healthy. And so Ty will tell you a little bit about some of the research that we're doing in this area towards the end of the presentation. So what are algae? What's the first thing you think of when someone asks you, okay, what's an algae? So you may think about this. This is kind of the most obvious one. It's kelp. If you go out on the beaches, you see this washing up on shore. Kelp are large. They're as large as plants. Some of them are as large as trees. They're also called, from the scientific perspective, they're called macroalgae, macro for large. because they're big enough to be seen with our naked eyes, right? Not without a microscope. Kelp are incredibly productive organisms. They can grow as fast as 2 feet per day, which is really much faster than any plant that we know of. And so they're really amazing. But actually, most algae are not macroalgae. Most algae are small. Here's an example of one that you may have heard of. You might have heard of bioluminescent algae. These are glow-in-the-dark algae. Most of them are called dinoflagellates. You can actually order glow-in-the-dark algae from the internet. So you can look that up and maybe order some and play with them. They glow in the dark when they're shaken up, when either the waves or fish run through them. And this is believed to be an adaptation to predator avoidance. You can see actually bioluminescent algae if you go look at the red tides in Southern California that occur oftentimes during late summer and fall. Another part of algae that you might have heard of are harmful or toxic algae. So these are so-called bad algae. You may have also heard this referred to as HAB or harmful algal blooms. This is what happens when algae grow to really, really high abundances, and the particular species of algae that cause these issues produce toxins. These toxins are bad for wildlife, for fish, for animals eating the fish, and also for humans. And there's two different kinds of those toxic, harmful algal blooms. There are some that grow in fresh water, so in lakes and rivers, including the Great Lakes. And then there's also the kind that grow in the coastal environments in the Pacific Ocean, the Atlantic Ocean, and the Gulf of Mexico. And these when these algae bloom, they cause toxic events, and shellfish accumulate that toxin and can make us sick. So fortunately, the state is very good at monitoring for these toxins, and so seafood is always safe, and the state tests the seafood for these toxins, and so as soon as they detect the toxins, they shut down the fisheries, and none of that material gets to our tables. And so this is an example where science is really helping us keep us healthy. And so most algae, as I sort of briefly mentioned, are plankton. They're very small. They're also known as microalgae, micro for small. And so you need a microscope to look at them. So if you look at some algae or a plankton sample in the microscope, you can see there's a variety of colors and shapes and sizes. But they all have one thing in common. They're algae. And we'll get to what that thing they have in common in a second. But what are algae good for? Are we already using algae for good things? Well, yes, actually, algae produce compounds that are really useful to us. You can buy nutritional supplements, for example, omega-3 fatty acids that are made by algae. You can buy beauty products that are made from algae. They can make very specific kinds of compounds that are useful for us. Another way that algae are used for good is to clean up wastewater. So wastewater is very high in nutrients like phosphorus and nitrogen, and algae are really good at taking up those nutrients and removing it so that the water that comes out of these wastewater treatment plants is clean. And in addition to that, algae in the oceans and lakes sustain our fisheries. So the entire ocean ecosystem, from fish to whales to everything in between, is all sustained by microalgae. And we'll talk about how that works in a second. So let's get back to the role of algae in the global scale. What do they do? Why are they so important? Well, they're key organisms in what we call the global carbon cycle. So what is the carbon cycle? You might have seen the carbon cycle in school and even on the internet, and it's sort of very complicated. There's lots of arrows going in and out and things like that. So we're going to simplify it a little bit and think about first the biological carbon cycle. So all life on Earth is based on carbon. So you have a lot of carbon in you, and all organisms do. And then carbon's also found in soils. Carbon's found underneath the ground in these carbon deposits that you can see kind of on the bottom of this cartoon here. And carbon is also found in the atmosphere, mostly as carbon dioxide or CO2. And so the biological carbon cycle sort of goes like this. CO2 comes into the land. It's taken up by trees and grasses and plants. And then animals or herbivores eat those plants. And then predators eat those herbivores. And then throughout this whole cycle, you can see the arrows are going down in size, right? So the bigger arrow from carbon dioxide into land, and then smaller arrow into herbivores, and even smaller arrows down down. And that means the reason for this is that there's a lot of carbon that's recycled through this process that goes back up into the atmosphere. So as carbon cycles through the biosphere, only a very small amount of it gets buried underground. And this is called carbon sequestration. So this is a process that happens year over year over year, and over thousands of years and millions of years, that carbon gets buried underground. Okay, so this is a process that takes a long time. But this is only half the story. The other half of the story, of course, is the ocean, water, because of course we live on the blue planet. About 70% of the surface of our planet is covered by the oceans. And so the carbon cycle in the ocean is essentially the same as on land. We have carbon dioxide going into the ocean. It is taken up by plants. In this case, they are microscopic plants called algae that we've been discussing. And then organisms eat those plants, and bigger organisms eat those plants, et cetera, et cetera. And eventually, over millions of years, just like on land, carbon gets buried or sequestered underground. Okay? So what is the process that leads to this? Well, it's photosynthesis. You might have heard photosynthesis in the context of plants. Plants and algae both do photosynthesis. That's what they have in common. In fact, a lot of algae are descendants of plants. So green algae eventually became land plants. So they invented this process of photosynthesis about 2 billion years ago. Before that, there was no oxygen in the atmosphere at all. So we couldn't even have lived 2 billion years ago. But when photosynthesis was invented, these organisms basically figured out a way to use energy, water, and carbon dioxide, and essentially make sugars, right? So in this here we have it shown kind of in equation form. So you need 6 molecules of CO2 and 6 molecules of water to make 1 molecule of sugar. In this case, this is glucose, so it has 6 carbons, and it makes oxygen. This is the process that essentially oxygenated the atmosphere, because before this process occurred, there was no oxygen in the atmosphere. Okay, so algae and plants both do this. And how does this actually work? How does photosynthesis put carbon into the ecosystem? Well, on land, trees take up the CO2 and release oxygen directly in the atmosphere, and they essentially pump sugar and other kinds of carbon underground into soil. In the ocean and lakes, it's the same thing, except it's mostly single-celled organisms, microalgae, that do this. So you have to look at them through the microscope. And at the single cell level, a single algal cell takes up CO2 and makes oxygen and sugars. So it's the same process, just at a different scale. And so let's take a look at how this works in terms of the ocean. We have this nice movie here that I'm going to play. And so we have the atmosphere and the surface ocean, where there are waves and things like that. And the CO2 goes from the atmosphere into the water. And then the algae are in the water, and they're photosynthesizing. And then little organisms called copepods eat them. Right, here we go. They eat them up. And then larger organisms, this is the sort of the ecosystem, the food web. Then fish eat them. And then larger fish, you're going to see a real big tuna eat them, right? And throughout this whole process, I'm going to just pause it right here. Throughout this whole process, we lose a lot of carbon through sinking. So dead algal cells sink out, dead fish, dead whales even, which is not shown here, sink out. And I'm going to play it again. And you can see this stuff sinks from the atmosphere, from the surface ocean, down into the bottom of the ocean. This is also called marine snow. You might have heard of this. And this is how it gets buried over time, right? Every year, all this stuff gets buried at the bottom, and it gets covered by other things. And over millions of years, this material pressurizes and becomes oil deposits. Now let's take a look again real quick at what's happening at the surface, because there's some nuances here. Oh, let's move to the next one. Okay, so here, take a look at the surface there. You can see that there's the carbon dioxide coming in. You can see that, and you can see the marine snow sinking. We're playing it again. So we see carbon dioxide coming in at the top, marine snow snowing down at the bottom. But then you just saw it right there. There was a burp. Okay, we're going to watch it again. There's some bubbles coming back up from the bottom, and it's going to burp right here. There you go. So you just saw the ocean burping. Okay, so essentially what this is meant to show you is that there's carbon moving both from the atmosphere into the ocean, and then occasionally it moves from the ocean back to the atmosphere. Okay, so this is what carbon cycling at the ocean scale really means. Okay, so here's what this means on the global scale. About half of the oxygen that you're breathing right now was created by microalgae, and the other half was created by plants. And so here what we're showing is the seasonal cycle of photosynthetic carbon fixation, essentially movement of CO2 from the atmosphere into either land and the ocean. So if you first look at the land scale, you can see the green kind of moving up and down with the seasons, right? In the spring, you have lots of trees, lots of green grass. We have carbon dioxide moving from the atmosphere into land. And in the summer and fall, trees start to lose their leaves, right? Grasses turn yellow, and then you actually have less carbon moving from the atmosphere into land. And then you see the same thing actually happening at the ocean scale. So if you follow the green on the land, it kind of also follows the same pattern as the oceans. And so again, this is this seasonal cycle of carbon dioxide moving from the atmosphere into land and then being buried, like we just showed you in the movie. And over millions and millions of years, this material again settles down underneath the ocean or underneath the ground and gets buried or sequestered, right? And in the ocean, this is called the biological carbon pump. This is the way that the ocean pumps carbon from the atmosphere to the surface ocean and then down deep underground. Okay. So now we're going to switch over to our high school demo with Erin McKay and her student Sarah.
Thank you very much, Xavier. So welcome to Tracy High, everybody at home. And I'm here in my biotech classroom, and I'm joined here by my student, Sarah. Sarah, come on in. Come a little closer. We know the microphone's a little picky. So we have been working on an experiment here in biotech. And as we've been doing our experiment, we have been playing with some algae. So let me just pull up the algae so people here can see. And let me just give it a nice little swirl. So Sarah, what do you observe?
Well, obviously there's a bunch of tiny green orbs, but if I had to guess, I'd say that there's probably millions of algae inside each little orb.
Yes, you're right. There are many, many algae inside each of those orbs. They're little gelatinous globules. They're like a world of algae, and we put them into an ocean. And instead of looking at the seasons, we're looking at the days. So first of all, let's take a look here at the first part of our experiment. In our first part of our experiment, it's just a bundle of foil. Oh, wait, is it a bundle of foil? Let's open it up. So if we take a look here, let me just give it a quick shake. So what color does the ocean that these algae are in look?
Well, it's a light yellow color.
I wonder why it's light yellow. So what does the foil represent?
The foil represents a lack of light, which represents night.
So yes, these algae have been in a simulated nighttime. And then we have some other algae in another container in a simulated foil. And let me just hold this a little closer for just a second because it's a little dark and take it back. So what color do you observe here?
A dark purple color.
And where were these? Were these in the dark or were these in the light?
These are in the light.
So what do the algae do when it's light?
They do photosynthesis.
And when they do photosynthesis, what do they make for themselves?
They make sugar because they're very hungry little algae, but they also make some oxygen.
And when they're making the sugar, what do they take out of the ocean water?
Carbon dioxide.
Yeah, so what color tells us that there's little carbon dioxide in the ocean?
The purple color tells us that there's only a little carbon dioxide in the ocean.
And
the yellow color tells us that there's lots of carbon dioxide in the ocean.
Can you hold them up side by side so everyone can see at home a side-by-side comparison? So there we go. We can see on a daily cycle what happens and how the algae use carbon dioxide that goes into the ocean water to do photosynthesis to feed themselves and provide us and every other living thing with oxygen. Thank you very much, and back to you, Xavier.
That was fantastic, Erin, Sarah. Thank you. That's a really fun experiment. All right, so now we are back to talking about the carbon cycle. Let's wait till we get to full screen. Awesome. Next slide. And so we talked about how CO2 goes into both plants and microalgae. And over the past 150 years, starting 150 years ago, something else has happened. That's we have kind of come along. We've been around for a long time, but over the past 150 years, we've started really using a lot of energy generation. To do that, we've had to pump carbon down from underneath the ground, both under land and under the oceans. And we burn that carbon and make up lots of energy, obviously, to ride our cars. Now we ride on airplanes to power our iPhones and our computers. And this has led to increased carbon dioxide in the atmosphere, right? And the reason for this is that we are producing about 10 petagrams of carbon per year. And plants are able to absorb some of that. They're absorbing about 2, and algae are absorbing about 2. But that leaves 6 petagrams of carbon per year that's just accumulating. How much is that, though? Like 1 petagram, what is that? So 1 petagram is the mass of 200 million African elephants. So the total amount of human activity per year is 2 billion African elephants worth of carbon. That is a lot of carbon, right? And so it's no wonder that the Earth is not able to account for all that, right? It's taken up about 4 back. So 6 petagrams of carbon per year are accumulating in the atmosphere as CO2. So if you go out and measure carbon dioxide in the atmosphere, and for example, in this case, this is the last 20 years, you'll see it increase over time. It's increasing fairly linearly, and you'll see some squiggles, right, every year. The squiggles, that's the seasonal cycle, right? Whereas during the spring, less CO2 accumulates, but then after that, it gets respired away and then more CO2. But overall, it's a gradual increase. And this gradual increase in CO2 is associated with an increase in temperature, as can be shown here. So this is because CO2 absorbs heat. So normally you have sunlight coming onto the Earth, and a lot of it gets reflected. A lot of that heat gets reflected out. But if you have more carbon dioxide in the atmosphere, it'll actually keep more of that heat in there. It's almost like a blanket. And so this is what's causing the temperature to increase over time. Next slide, please. And so, we are in the age of climate change. It's a major crisis. It's something we're going to have to deal with in my lifetime. You're going to have to deal with it in your entire lifetime. And so it's something that is no longer just an environmental issue. Climate change is an issue at everywhere. It's an economic issue. It's a safety issue. It's a health issue, right? And it's also an opportunity for us to really figure out how to fix the problem, right? This is something that we all should care about. And so, Lawrence Livermore National Lab and the government, the lab system in the United States is also part of the solution. We have to figure out a way to fix the problem. Next slide. And so we think that algae are part of the solution. So, we could already use macroalgae, either grow them in seaweed farms and then dry them, or we could grow microalgae in like microalgal farms, as you can see on the top. And you can collect this material and actually make useful products like plastics and things like that, but also biofuels. And so this is what we're going to talk about for the rest of the talk. And we have, before Ty takes over, we have a little movie that was produced just a couple years ago, so pre-COVID, so there are no masks, that is going to introduce you to the kind of research that we're doing at Lawrence Livermore in this topic.
When I say the word algae, the first thing that may come to mind for you is something like the swamp monster. I mean, algae is just plain old pond scum, right? Well, recently, Lawrence Livermore National Lab was given a $3.5 million grant to keep working on scientific projects about algae. But why? What about algae is so important? Today, we're going to talk to 3 scientists and learn what algae is, why it's important, hint, it can be used as a biofuel and to capture carbon, and how we're collaborating with other labs to bring algae technology to reality. Now, let's go learn about some green squishy stuff.
Rona, what is this lovely flask of greenness?
So these are called microalgae.
Okay.
So microalgae are single-celled organisms. They're aquatic usually, they grow in water. Algae is a big group. There's a bunch of different organisms that are included in it, but they're all sort of single-celled organisms, and the thing they all have in common is that they photosynthesize. Actually, these green algae here, these are green algae, this is Nanocropsis, and this is a direct sort of plants, all land plants came from green algae.
Oh my gosh, wow, so we're talking to someone very important here.
Yes.
Okay, so why do we care about algae? What's important about them?
So first of all, we care about them because they're very important to our planet, right? They're a base of the food chain and a lot of ecosystems in the world, and the oceans, sort of everywhere, they're the base of the food chain. But we can also use them for cool things. So for example, we can use them to make fuel. They naturally make lipids, which we can make into biodiesel and just put directly in our cars.
Whoa, like my car that I have outside that's just like normal? So in theory, you could have this wonderful sort of cyclical loop of feeding algae CO2 and then having the algae be a fuel.
Yes.
And you could even use recycled water.
Right.
So this would be part of like a very sustainable
Yes, that's
cycle.
the whole idea, is that they can be sort of a sustainable biofuel product.
Sure.
That they're not bringing more CO2 into the atmosphere, they're taking it out, then you burn it and put the same back in. But unlike fossil fuels that have been buried for millions of years, this isn't extra CO2. So it's carbon neutral.
It's all being sort of encapsulated in this one loop.
Yes.
Okay.
Exactly. So you take out the biofuel, but you can also take the rest of that biomass that isn't the lipids and the diesel that we want, and you can use that for animal feed, let's say.
Okay.
So you can use it as fertilizer. So some
nice.
people are experimenting with that. It can also be used for human consumption, so that's already. So some spirulina, which people may have heard of.
That's algae?
That's algae.
I had no idea.
Yeah, and the other thing is you could, because algae are these tractable organisms that we can genetically engineer, you can also have them make really whatever you want, any bioproducts. They can make insulin or something like that. That's a, a compound that we usually have E. coli make.
Right.
But here you could have an algae make it so that you get
might be a little
diesel,
more manageable. Yeah.
So at Lawrence Livermore, we're interested in collecting algae and growing them. But the current issue is that when we grow them right now, they don't grow very well. And so the prices for them are very expensive. So our goal is to improve their growth such that prices can decrease. And then in doing so, we can create more algae that creates our own pool of carbon. That when we burn that and turn it into fuel, after we turn it into fuels, the combustion by cars and transportation vessels and commercial airlines, what that does is that CO2 then enters the atmosphere, and then the algae are able to reabsorb that CO2. And so you can see that it's more of a cycle. So the algae are reabsorbing that carbon, and then the process continues again. The algae biomass is extracted, turned into fuels and products. It's burned or turned into CO2 in some other way. It enters the atmosphere. Then the algae reabsorb it in these algae ponds. So this is an example of sustainable bioenergy, and we really need this for the 21st century because of what Xavier was mentioning. And this is just a recapitulation of what I was just talking about. The algae absorb carbon dioxide and water and sunlight through photosynthesis. It's absorbed, and then we can take that biomass, refine it, turn it into oil, burn it, and then the carbon dioxide goes back into the atmosphere. And so this whole process is carbon neutral because it's not a one-way movement of carbon from deep in the earth in oil deposits into the atmosphere. It's moving into the atmosphere and then it's being reused. So again, it's sustainable and we're recycling it, so it's carbon neutral. So some of you may be wondering, how do we grow algae at LLNL and folks in general, how do they do this? At Livermore, we have these raceway ponds, and so you can see the algae growing in here. There's a paddle wheel that is mixing the algae up to make sure that they're able to get sunlight and nutrients. And in these little raceways, we have sensors that are measuring temperature and the amount of CO2 that they're absorbing so that we can be sure that they're receiving enough CO2 and being able to grow. And then all of that is routed into this little array that we can pull the data from and take a look and see what the results were. I'd like to introduce our algal cast of characters, and Rona talked about a little bit of these in the video, but we work with just a small group. Diatoms, which are golden algae, maybe some of you have heard of them. Phaeodactylum tricornutum is its official name. We also work with green algae, and green algae are the progenitors for all land plants, and they all have chlorophyll. So they're performing photosynthesis, and they're turning those into compounds and releasing oxygen. And those compounds can be turned into fuels, but then they can also be turned into materials. There's some research that's looking at turning various compounds into yoga mats and flip-flops, which is pretty neat because when these things are discarded and they make it into landfills, they don't break down very easily. So being able to turn these things into something that can be recycled or comes from renewable sources is ideal. So algae are very small, and so they make for big challenges. Here I'm showing a milliliter centrifuge tube, and 1 milliliter is 1 centimeter by 1 centimeter by 1 centimeter, right? So that's a cubic centimeter. Some of you might know of a cubic centimeter as a cc. And so if you can visualize this amount of water and think about human hair, I don't know if you have a human hair lying around, but yeah, take it. And if you put it in like that cube of water, this is about the size that it would be. And then one algal cell relative to that human hair is super tiny. So I'm not sure, really get in there. I have a little flashing algae in there. I don't know if your resolution is good enough or if you're looking at this on your phone, but it's there. And so we'll zoom in on that and we'll take a little bit closer look and see just how small these algae are. And we can see when we do that algae aren't just small, they're in fact microscopic. So here's that human hair, and here comes our algae zooming in. So if we look at it at this scale, we can see that on average about 3 algal cells can fit across a human hair. So if you look at your human hair on end, 3 algae can fit across that diameter. So just minuscule, super, super tiny. And the reason they're able to have such an impact on global climate is because they're so numerous and they're growing so fast. So these little tiny things are super important because they can grow so fast and they're just extremely numerous. Some of you might be wondering, how do we look at things that are this tiny? And some of you might be already screaming into your screen that you know the answer. And you are probably right. We use microscopy. So we collect our sample into our little wells here. We put the well onto the stage. We move the stage around so that it aligns with the objective. The objective is what we use to magnify our image. We make sure that we have a little bit of light to generate contrast. And then we have to adjust the focal plane to make sure that our algae are in focus. Make a couple more adjustments. And then this particular microscope, we can actually port the image through a camera and then visualize everything on a computer. And so what I'm doing here is I'm gradually zooming in. So you can see the little scale bar. That white bar is 50 microns. And these are those diatoms that I showed you earlier. And they're autofluorescing red. So this is their chlorophyll. When you hit it with blue light, they fluoresce red. And then I'm zooming in here. You can see the scale bar now says 10 microns. And then we'll switch over to this chlorophyll signal, and we can see the chlorophyll as little dots within those cells. And so that's where all the action is occurring, right? That's those little organelles, those little features within the cells. That's where photosynthesis is happening, where the CO2 is being incorporated and being turned into sugars and lipids and proteins like that, and then the oxygen is being released from those cells. So what can we do when we see these microbes? We can see how interactions are occurring either by taking videos or watching them in real time. And we can figure out what the problem is if there's an algal culture that doesn't seem like it's growing very well. And then we can figure out if the treatments that we're applying are effective or not. So in this case, you can see in the blue, these little blue cells they're called chytrids or chytrid fungi. And what they do is they attach to a bacterium, insert this little thing called a rhizoid, and then basically suck the algae dry. They eat the chlorophyll, they eat all the yummy stuff within the algae. And in this case, they're infecting these algae called Synodesmus. And this is bad if you want to grow algae outdoors and generate a lot of fuel and products, plastics and stuff from that biomass, because you don't want the chytrids to take all your biomass. You want to take that biomass and refine it into something that you can use. But this is also interesting from just an environmental standpoint, because this is happening in real environments, in lakes and streams, in the ocean. And it's just part of that natural carbon cycle that Xavier was talking about. In this case, the chytrids will be eating the algae, and then maybe those chytrids are being eaten by something else, and then it's a different type of food chain that's operating. So it's interesting from both standpoints, from the environmental and then also from like the applied biofuels side. So you may be wondering what predator-prey dynamics have to do with microbes, but it turns out that many of the interactions that we look at are playing out like this. So in this case, we have the hare and the lynx, and we can see that over time there's this little oscillation that happens. The hare populations increase, And then the lynx populations increase because they eat the hare, and then the hare populations go down. And then there's no hares for the lynx to consume, so their populations go down. And without the predator, the hare population goes up. And so this happens everywhere. This is happening with deer and wolves, for example. But the same thing is happening at the microscale. These concepts are interchangeable. The only difference is that we can see this with the unaided eye, but with microbes, we need help to see that. So here's an example of that occurring at the microscale, these predator-prey dynamics happening. But in this case, we have our algae and we have ciliates. So in this case, what do you think is the predator and what is the prey? So we're gonna put a little poll up for you, and you can vote after watching this video, or maybe you already know. Maybe this is just known to you, but let's take a look, because this is a pretty interesting video. Oh, and also I will not be telling you the answer. We'll make our way through this together. Oh yeah, look at that. One more time. All right, got that? Cool. So using microscopy can reveal other secrets about this fascinating microbial world. In this case, it can tell us the degree to which other microbes might be helping algae. So here we have the microbiome, an image of the microbiome interacting with Nanochloropsis. And the microbiome is just comprised, it's a fancy, easier word to say than bacteria or bacterial community. So all these little dots are individual bacteria that are attached to and interacting with this Nanochloropsis algae. And taken together, all these little dots, we just call all those little dots together the microbiome. And we can see here that they're attaching to each other, they're interacting strongly, and we can use microscopy to see how those interactions change with time. We can go in and we can measure how big the cells are, or how many cells are attached to one another, things like that. And then that informs us how these interactions play out and how we might need to manipulate them to make the algae grow better. This cooperation between algae and microbiomes brings us to another important concept in ecology that we can illustrate with this macroscale example, and that is that symbiosis requires proximity. Here we have a shark. Many of us who have watched nature documentaries have seen that sharks are frequently associated with remoras. And these remoras offer an important service to the shark. They're cleaning parasites off them and just skin. They're eating skin. They're cleaning the teeth around the shark, getting excess food, make sure there's no infection or anything like that. And then in return, when the shark feeds, that those remoras receive the excess food that sharks aren't known for their table manners, so there's food flying everywhere, and the remoras get to eat that. And so this is an example of a mutualistic symbiosis, but it is necessitated by this close interaction and this close proximity. So this extends to the microscale as well. And in fact, attachment between algae and their microbiomes is critical for these algal microbiome symbioses. So here I'm showing you two groups of bacteria. One are the attached cells that are here in pink, and so they adhere to the algae. And then there's also within these cultures there are unattached cells. They are what we call free-floating or motile. So they're able to move around, or they're just kind of stuck, and they rely on interacting with the algae by just kind of bumping into them. And so all of them can help the algae in some way, potentially. But we know that because the attached ones are right there with the algae, they are the ones that are interacting most intensely. And so we have a real life example here. Here's some example data. So when we grow these algae under optimal lab conditions, under normal light and temperature, we can see that microbiomes are helpful. You can see on the right there, the algae with the microbiome are bigger, and each of those points are a single cell that we've observed on the microscope. In contrast, though, when we grow these algae under high light and high temperature, which simulate outdoor summer conditions, we see that microbiomes are actually not helpful, and the algae cells are actually smaller. And the reason we're looking at these and comparing these is because when you grow algae outside, it's best to grow them where in high light, where they're not light limited because they need light to photosynthesize. But the best places for that turn out to be also be the hottest places like Arizona or in the California desert. So we need to figure out how to optimize algal microbiomes to basically work in these conditions such that they can help the algae grow and even think about protecting them from light or other types of stresses. In this case, it's interesting that we don't see the microbiome helping the algae because this suggests that there's some kind of imbalance happening such that the bacteria in that microbiome are actually harming the algae, right? So the algae are actually smaller. And this may be because the good bacteria have been killed by the high light and temperature, and so it's only the bad ones that remain, and then they're coming and attacking the algae. Or it could just be something as simple as there's different compounds that are being released by the bacteria that harm the algae. There's all sorts of hypotheses that we can then go in and investigate further in the lab, and that's kind of the essence of science. There's always more questions. So I think we've illustrated to you today that LLNL uses observations to study algae, but as I just alluded to, this is just one of many tools and expertise that we need. And so if you're interested in this and you're thinking about what you want to do, as you move on through college and maybe grad school or whatever, and if you're interested in this, we need expertise in genomics, things like genetic engineering, chromatography, which is involved with chemistry, and mass spectrometry is another example of a chemical skill that we leverage often in the lab. And I'm showing that an image of a type of mass spectrometer that we use at Lawrence Livermore National Lab for this work. This is called a nanosims, and the SIMS stands for secondary ion mass spectrometry, and that's just a fancy way of saying that we're able to look at how the algae and the bacteria are exchanging compounds, and we can see it happening with this awesome instrument. And then mass spectrometry can also be used to look at what compounds are being exchanged between organisms and in other ways and things like that. We also need expertise and skills in material science to figure out if there are ways that we can encapsulate algae and microbiomes and have them interact in different ways. And then all of these approaches are just generate loads and loads of data. So we need skills in programming and coding in order to parse out that data and figure out if there's any patterns or if there's ways that we can simplify the data to get a better understanding of what is happening. So some takeaways from the things that we've discussed today. Algae are mind-bogglingly small, and we need microscopy in order to observe them and make these observations that lets us move forward with testing hypotheses in the lab. I've shown you just a small subset of algae, but there really are thousands, probably millions of species that live in oceans, lakes, streams. There's even algae that live on ice in the Antarctic and in the Arctic. And they're crucial for the health of the planet because they photosynthesize. They are the reason we can breathe. Every second breath you take, remember to thank algae. Just kidding. That would be tedious, but you get the idea. And also because of CO2, because of photosynthesis, they're able to recycle CO2. And this enables us to generate fuels and materials from marine algae and then regenerate them because we're able to recapture that CO2 through algal photosynthesis. And at Lawrence Livermore, we are specifically looking at microbiomes that attach to algae and increase their growth a unique approach to make sure that these interactions are robust and strong through time and in different environments. We're using a variety of tools at the lab to look at this and start making progress on really improving algal growth and being able to scale that up into the large scale and start generating more and more renewable, sustainable fuels and materials. And with that, I think we'll move on to questions.
Thank you, both Ty and Xavier. That was a great job. So let's go ahead and look at some of the questions that I've seen in the chat. So let's go ahead and start with one of my favorite questions that I always have to address in class from middle school to high school, which is, are all plants in the ocean algae? So plants and algae, what's going on there?
Ty, why don't you take this one?
Okay. So I mentioned on one of the slides that green algae are the ancestors to all land plants. And so when we're talking about ancestry, we're talking about evolution, right? So algae at some point started becoming multicellular, and they started doing that in the ocean. Now, the algae that became multicellular and stayed in the ocean, a lot of those are considered algae, whereas some evolved further and they're becoming more like plants. So it starts the lines between plants and algae kind of start blurring, but in general, most of the things that photosynthesize in the ocean could be considered algae, but there are some that are more plant-like for various reasons, and there's a whole scientific discipline that looks at that. So it's a really good question, and it's actually it's a field of research in and of itself.
Great, thank you. And another question that people had is just what else can algae make? So can we just get a quick rundown of the interesting things that we can use algae to make?
Yeah, so algae make lots of protein. So you can make protein extracts out of algal cultures. They make lipids. That was sort of in the video where Rona talked about that. So algae can actually make oil inside of their cells, and then you can extract those oils and use that for biodiesel, for example. And then algae make all kinds of vitamins and nutritious compounds, very expensive compounds actually. So the companies right now that are growing algae and making money, they do not grow them for biofuels because it's too expensive. Right now, we cannot compete with the price of fossil fuels. But the ones company that do exist, they will make nutraceuticals, so very expensive compounds. Usually they're related to pigments because the reason algae are so colorful is that they have all kinds of different pigments that absorb light of different wavelengths and allow them to do photosynthesis. And these compounds actually can be very useful chemically, and they're expensive to make through chemistry, so they're a lot cheaper to make if you just grow algae and then extract the chemical from them.
Thank you very much. And there's another question, and I know it's related to the worksheet, but it'd be great to just hear from Ty. And it's just related to your video when it was showing you working with the microscope. So Ty, can you just give us a quick rundown what it's like when you're working with your microscope and what you like about it and what you get to see?
Yeah, so you might have sensed that microscopy is one of my favorite things to do in the lab. And in general, my favorite thing to do is just that first look that the video captured. You have your sample and you have it in the right focal plane, and all of a sudden you're just exposed to this new microscale world, and microscopes are so good nowadays that you're almost able to look at the microbial world from a microbial eye view, right? They don't have eyes, but you can start getting a sense of how they're interacting with each other. A lot of these bacteria, they're motile, so they'll come and you'll see it, and you can tell that they're just kind of around there sensing the environment, and then they'll scoot off. And just being able to observe that and see it is just always incredible. to me. And some other projects that we're doing at Lawrence Livermore is trying to better understand what is happening when those algae are coming by or sticking. And so we're trying to look at things like how is the local oxygen concentration changing? How active are those bacteria? So are they pretty healthy, or are they there because they're trying to become more healthy? So we're trying to figure that out, because if you have a healthy microbiome, then you potentially have healthy algal cells. And so we're being able to just observe these types of things and not have to take a sample, process it in some way, and then just be like, I think this could be happening. When you're actually seeing it, it's so much more interesting and powerful, at least for me. Hope that answered the question. I'm starting to get really excited. I could be here all day talking about microscopy. I'll tell you that right now.
And I could just listen to it all day. So, and actually you did a great job, because you not only helped with that, but another question we had in there, which was, why was it so critical that you guys at Lawrence Livermore National Laboratory are studying the microbiome and the interactions going on there? So you did a great job hitting that also. So just a little quick thing that I know a few students were just struggling with, because it just kind of went by quickly, is what is the percentage of oxygen that algae are helping produce in our atmosphere?
Yeah, I didn't answer the question. I was trying to answer the questions in the chats, but since it's a worksheet question, but you're right. I went over it too fast. So on average, it's about half. But what's really interesting here is that if you look at how much biomass there is, like if you took all the plants on Earth and like weighed them, and you took all the algae on Earth and weighed them, they're completely not equal at all. There's way more plants, okay? Because trees are really big, they have a lot of right. So in terms of mass, it's much more plants than algae. However, in terms of activity, it's equal. And the reason for that is algae are, number one, really small, and they're also really active, and they're constantly being eaten really fast by other organisms. So it's cycling much quicker, whereas on land it's cycling much smaller. So you have basically two different kinds of ecosystem that produce the same amount of oxygen. One, you have high biomass and low cycling, and the other one you have low biomass and high or fast cycling. And that's kind of the difference between land and ocean. You can think about it in this way. And I will say, I think there are a lot of really cool questions in the Q&A. I don't think we're going to get a time to answer them all. I'm willing to stay on here after the hour's over and answer them by chat, but I don't know if that's possible to leave that up. But I just wanted to mention that. I haven't been able to answer all of them so far.
Well, I've just been watching what Dr. Albala has been saying, and we have time for a couple more questions. So just along those lines about the plant versus algae, how about there's another question in the chat of, are there more microalgae or macroalgae in the world? And I'll leave it to you to whether talk about biomass or biodiversity or both.
Yeah, interesting. Ty, you want me to take that, or you want to take that?
Yeah, go for that. Yeah.
Yeah, so in terms of biomass, for sure, there are more microalgae, the small ones. That's just because macroalgae, the big ones, really are only able to live in the very close environment, close to the land, because they have to be attached somewhere. So they can only live where there's there's only maybe about 20 meters of surface height. Otherwise, they're not going to get enough sunlight, right? So basically, most of the ocean is deep, is not shallow. So most of the ocean have microalgae. So even though they are smaller, obviously, they're just more numerous because there's so much more volume of water where the microalgae live compared to the macroalgae. In terms of biodiversity, probably also microalgae. So there are only a limited number of major algal families that have evolved to become macroalgae. So there's kelp and there's red algae. So red algae are like nori. If you eat sushi, that's nori is a red alga actually. And then the ones that you see most on the coast are kelp, and they're related to diatoms. They're actually related to the organism that Ty talked about, the phaeodactylum, the golden brown algae. But they're not red algae. But in terms of the biodiversity of microalgae, it's amazing. It's more diverse than plants and animals combined together, right? So it's huge amount of diversity of microalgae.
Okay, great, thank you. And there's another question as Ty was going over the talking about light's impact on the algal growth. And so the question was, does that mean that temperature affects algae also?
Yes, definitely. Um, it's we haven't been able to look at that in detail yet, but there are everything, almost everything on the planet has, temperature optima. Uh, Humans, even if you're in a room and it starts creeping up to, 70, 77, 78, you start getting, a little uncomfortable. Algae are the same way. Some like it much colder because that is the environment that they were naturally found in, whereas some, for example, those that are found, along the equator, they may like it much warmer. But almost pretty much everything on Earth doesn't like it when it gets too hot. And so if you can find ways of dealing with that or workarounds in terms of engineering the microbiome to help the algae in other ways, then you can deal with that. So just to provide a larger scale example, if you're on the savannah and there are zebras. The zebras in this case would be the algae. If they can find some shade, that's helpful. And one thing that Xavier and I have been trying to look at is there a possibility that microbiomes can provide shade to the algae? You can imagine if the bacteria are actually attaching to and dotting the outside of the algae, they're able to actually absorb some of that intense sunlight and maybe help the algae cope with this environmental stress.
Great. Another question that kind of came in multiple iterations that I had also seen that Xavier kind of addressed, but let's kind of look at it all together was, are bacteria both good and bad, and are algae both good and bad? Because there's all a bunch of questions related to that.
Yeah, absolutely. I mean, it's all on how you look at it, right? So we have to look at it from the point of view of humans, because of course, from the point of view of algae, there's no good and bad. There's just survival, right? But so from our point of view, the algae that produce toxins, we consider them bad because they cause us problems. They contaminate our seafood, right? They kill fish. The dead fish wind up on our beaches, and then they smell bad because they're decomposing, and then nobody's happy, right? The people tourism industry is unhappy, right? So those are the bad algae. Most of the time, obviously, most algae are good, right? They are like we mentioned, this whole time, they're producing oxygen. Without them, we couldn't live. And of course, they're helping mitigate the increase in CO2 in the atmospheres as much as they possibly can. So in most cases, they're good. In terms of the bacteria, it's the same thing. It's all about perspective, right? So bacterial diseases obviously are bad for us. And there are bacterial diseases of algae. So from the point of view of algae, bacterial diseases of algae are bad, right? But most algae, in fact, are good. Most algae are part of the natural ecosystem. They degrade dead organic matter. They recycle carbon and nitrogen and phosphorus so that the ecosystem can continue to grow. So in fact, without bacteria, our world would not even work at all, right? And so it's all in perspective. But from the point of view of from the perspective of humans, most bacteria are good, most algae are good, only a very few bad actors are bad. So I think algae and bacteria both get a bad rap, right? Because a few of the bad ones get all the notoriety, but most of them actually are good.
And so just our final question is, how much algae are needed to recycle one person's breathing?
Wow, that's a great question. I don't actually have the answer to that, unfortunately. I would have to do a little bit of research to answer that question. That is a fantastic question, right? Because it's all about numbers. And yeah, I unfortunately I can't answer that. I don't know if Ty, if you have that info, but
No, yeah, that is a great question. I think it depends on a lot of things. Not everyone respires the same amount of CO2. But yeah, great question. I've always had this dream of just hooking up like some culture to the tailpipe of my car, and then just having that CO2 from my car just go directly into like an algal culture. And I wonder if the person who posed that question is kind of thinking something similar. If occasionally we just walk around in bubbles and respire and support algal growth. I don't know. That's maybe that's a little bit too sci-fi, but yeah.
It just goes to show that we can even come up with questions that the lead researchers still need to dive further into. Great job, guys. And I'll throw it back to Dr. Albella.
Well, I just want to thank everybody for a fantastic presentation today, for sharing your passion for your work, your research, and our planet. And I'd like to just share that we'll try, if it's possible, to get some of these questions answered and put it on the education.llnl.gov site. May take us a little while. So if you need to turn those worksheets into your teacher, don't wait for us. But there were some really fascinating questions that I'm sure you'd like to get the answers to. And perhaps you can start a conversation with your science teacher at your school as well. And please join us next Saturday for a fascinating look at fusion energy and the National Ignition Facility. Thank you all so much for joining us today, and have a wonderful weekend. Take care. Goodbye, everybody.